thanks for the intro, and thanks for coming to my talk today. So I'll be describing a little bit about advanced microscopes and also about signal processing needed to deal with the large amounts of data that comes out of these microscopes. So So yeah, every neuroscientist starts the talk with a Ramona Call drawing. So this is one of mine. So this is kind of the depiction back in the early 1900s of what the brain kind of looks like. And this person won the Nobel Prize for making these exquisitely detailed drawings of neural structure. So neuroscientists generally like to figure out how the brain works by looking at both structure and function. And an aspect of structure, people have heavily engineered this, right? Now people take very detailed images using electron microscopy and can derive the full kind of morphology to look at the different types of neurons, whether they be large ones that extend over multiple layers of the cortex or small ones that kind of operate over a smaller regime. Okay. So neuroscientists also like to look at function. So traditionally people have examined neural function by actually poking neurons with pipettes, making actual physical contact. So this is the first neuron I ever patched in Slice, but you put it on there and you can measure the action potential of these spikes of activity that are integral to the communication within the brain. Okay, so this is kind of how traditional neuroscientists do it, and in a heavily engineered way, this is what modern neuroscientists do. So this is from season 4 of Next Generation in Star Trek. The idea was that this person needed to interface himself with the computer, and he built this optical interface where lasers shoot into his head and carry information in and out. And honestly, this is not so different than what neuroscientists do nowadays, just like how Professor G showed in the last talk. Basically, there's an array of various types of protein indicators or other indicators that give flashes of light whenever there's neural activity. So these are partially developed in my lab, this one was, and this one is developed by Tenalia Farm. But the idea is that whenever there are spikes, there's concomitant calcium or voltage activity. And these activities are translated into these flashes of light that microscopes are trying to catch in a fluorescence configuration. Right. So, and that was on the small scale, each one neuron, and on the large scale, we can capture very large movies. This is 1 photon, so kind of blurry, and this is 2 photons, so very sharp, giving you the defined features of neuron cell bodies and their axonal projections. All right, so my lab works on kind of the amalgamation of these areas, and today I'm going to talk about optical microscopy in this field and signal processing in this field. So we'll start with optical microscopy. So as Professor G showed today and Professor Hillman showed yesterday in the symposium, the idea is that the brain is a 3D structure, so we need to capture volumetric information. The problem is that protein sensors tend to go all over the place. So they tend to emit light throughout the brain. And we certainly want to concentrate on one subset of those structures, like the cell bodies and this row stuff. All right. And there are multiple ways of doing that. The basic idea is to pattern the illumination so that we don't illuminate lots of stuff simultaneously. This can be done by light sheet, where a sheet of light can be scanned up or down, or this can be done by a variety of 2 photon, including the extended vessel beam Professor G just mentioned, that can scan laterally. And all of this stuff is meant to reduce the out-of-plane fluorescence, so we can just focus on one layer or one section of the brain at a time. And this can be done in zebrafish, and another example of this is shown here. Zebrafish is a nice model organism that's relatively optically transparent. So a lot of these optical tricks work really well in zebrafish because it tends not to diffract light. And you can see, again, some optical fluorescence activity while this zebrafish is awake and behaving in some manner. Okay, so all of these methods require some kind of scanning, and ultimately, in order to keep up with the calcium and especially voltage indicators, we'd like to get to a regime where the scanning speed is not necessarily the limitation of these microscopy volumetric imaging aids. So one way to do that that comprises the stuff I'm going to talk about is Lightfield microscopy. This was invented in about 2005, and the idea is that it differs from a conventional microscope by being able to image entire volumes within one frame of capture. All right, so how does it do that? So traditional microscopes use a main lens to image some kind of sample onto a fixed plane on the camera or sensor. Lightfield microscopes insert an additional microlens array into the microscope, and instead of imaging a classic kind of circle kind of shape, it images this weird thing. All right, without getting into too much detail, the light is split into small regions beneath the microlens arrays. But the important thing is that it can capture depth information simultaneously. And using computational methods, you can actually focus on different portions of the sample after the single frame is obtained. So in this case, one computational depth can focus at the top, which is this little region here, whereas another computational depth can focus on this region far away from the top of the sample. Okay, so this has been done already, and conventional light field microscopy essentially shoots light into the sample from above using epifluorescence, and then tries to try to figure out the depth information by capturing all this information back on camera. So the work that's been done by Defan Wang, a Calvin postdoc in the lab, has been to make new flavors of this light field microscopy. And I'll describe two very briefly today. One is called light sheet light field microscopy, and one is called structured illumination light field microscopy. So these two microscopes will respectively enhance the contrast and resolution of the light field modality. And I'll show you how it does that. So the first one I'll talk about is the light sheet excitation. So the basic idea is that we would restrict our excitation of the sample from the side using a conventional light sheet microscope and scan that up and down really quickly. So we can define a very targeted region of excitation using this volumetric light sheet excitation. And that's very beneficial because it reduces the out-of-plane fluorescence both above and below, just like how traditional epifluorescence has out-of-plane fluorescence, decreasing its contrast. This effect is very obvious when we image very kind of calibration samples involving beads. The top image is done using wide field illumination, so this bathes the entire sample in lots of light, whereas the bottom one is using this restricted light sheet approach. And you can see that although there are beads like the one above my cursor in this image, it almost gets washed out by the large background coming from the agarose that is actually relatively not fluorescent, but all of the background makes it highly fluorescent. Instead, when we use light sheet to restrict the excitation to just a very small volumetric range of depths, we can see that bead and lots of other beads show up in this image. In the end, the bottom image, when we quantified, had about an order of magnitude higher contrast just by removing all of the out-of-plane fluorescence. When comparing this to traditional light sheet microscopy, obviously this will have lower resolution, but it still captures qualitatively all the beads that light sheet microscopy would normally catch. And this is the second way where we made a very thin light sheet illumination and still image the light field. And so the basic idea is that we can capture about 30 to 50 microns of information while maintaining a decent amount of the contrast in the sample. We did this in zebrafish, and the same kind of qualitative notes can be seen again. So here's the wide field illumination. Basically, everything gets lit up in a large haze. And then in the light sheet approach that we did, we can see much more structure, including kind of the optic tectome. There's the thick forest of fibers, some cells, very much like how the higher resolution light sheet microscopy can do, except this required scanning, whereas this we can obtain some volumetric image within one capture of a frame. And we can find some calcium activity from these cells when we stimulated the fish auditory pathway during the gray bars. Okay, the second one that I'll briefly describe is structured illumination. So today, maybe we can just focus on me. I wore a very thinly patterned shirt today to demonstrate the incorrectness of doing this now that we're on Zoom. The idea is that you see this haze of stuff on my shirt, right? And that haze comes from essentially aliasing of a thin pattern on the small pixels of the camera. So this is bad for my appearance on the video, but it's actually very useful for looking at high spatial frequency information in a sample. So this haze is essentially this pattern of a thin stripe of my shirt against some pattern of pixels on the camera, okay? But it turns out if we can apply very high spatial frequency gratings into the sample, what that really does is that it can demodulate high frequency spatial information back to the baseband that can be captured by our camera, just like how the webcam is capturing my image right now. And this higher spatial frequency information leads to overall smaller spatial extent. This is what we learned from Transforms 101. Okay, so we're going to do that by essentially applying volumetric grading patterns into a sample. And we did that in a relatively clever way. So the idea is that we use the DMD to project a hologram, and the hologram was the interference pattern between two plane waves at the principal focal position of the sample. And this actually generates some volumetric grading pattern instead of just what people normally would think of as a one layer of grading pattern. This actually extends about 200 microns, which is not the limit of our microscopy depth range. Right? So once we did that, we again can image bead samples. Here's the traditional wide field microscopy, and you can see the bead samples being relatively blurry. Once we take multiple images with the higher spatial frequencies and do the reconstruction, we can use structured illumination to find much smaller bead sizes in the spatial realm. In the frequency realm, we see that the spatial ranges are actually bigger, so this is the inverse by, again, the transform relationship. And you can see that what used to be the limit of capture in our regular light field microscope is essentially doubled, and our spatial resolutions are correspondingly doubled, especially in the lateral direction. In the axial direction, we also had some gains, but as expected, it didn't do very much because the reconstruction was in the 2D plane. And lastly, we did this in zebrafish again. So this is more of the hindbrain, but again, you can see wide field microscopy gives a large haze of fluorescence, whereas the structured illumination can reveal finer features inside of the zebrafish hindbrain here. And again, we can do some imaging of the calcium response, and again, find the calcium response. So this higher spatial resolution also essentially allowed us to reject a lot of the background noise and allowed us to increase the SNR, which is pictured in these bar graphs, but I won't go into as much. Okay, so now that we have all of these movies of taking volumetric data, what we can do is also take we need to process it. So in the last section, I'm going to quickly go over the signal processing stuff in our lab. So basically, the idea is that we have these movies, and they contain hundreds or thousands of neurons. How do we go in and actually find them when those samples are heavily and densely labeled? So it turns out we're going to borrow from a lot of existing computational methods stemming from Google and Facebook, because what they're really good at now is finding particular faces and pictures. So this is what's known as object segmentation. You just take a still image and you can find a slow space. Or they can do action segmentation, where they can take a video of a skater and they can figure out whether this person is performing a foot sequence or doing some kind of jump. Okay, and basically this maps really well onto finding neurons in our imaging movies. So if we wanted to do object segmentation, we can take one image and we can have our algorithms find the individual neurons. And then if we had a movie, we can have some kind of algorithm find the active neuron as it's flashing as well. All right, so this began as a collaboration with Sina Farsiu in BME, and Samaya was the grad student who did this. She's since graduated. And in our initial attempt, we took advantage of deep learning. So all of the software is freely available now, and operating it actually is not that much of a chore. The general idea is that we need a human to label some set of neurons in these green markers, and then we can feed them as training samples into the neural network for it to chew on for many, many iterations. But as it chews on that, it can eventually learn what the spatial shapes are of the neurons, and it can spit out segmentations of what these neurons finally are. So our initial attempt at this, Samaya's innovation, was to use 3D neural networks. So instead of just looking at the spatial extent and looking at and learning about what the spatial shapes of the neurons look like, she also used a temporal extent in this 3D deep learning algorithm so that it can learn what active neurons look like. What do they look like when they're actually firing? The rest of this network is pretty complicated, but it's just to reduce the number of features for the neural network to try to learn on and actually spit out the neurons. So more recently, Yijun Bao, another talented postdoc in the lab, has been refreshing and coming up with a fresh look at this entire pipeline. So the pipeline actually looks very much the same. There's some pre-processing module, there's a inference module that tries to figure out where the neurons are, and there's some post-processing. So where is this innovation? So the key innovations that we made here was that we tried to lessen the burden on the deep learning algorithm. We know what neurons look like. We know kind of how big they are. We know what kind of temporal waveforms they have when they fire. So we integrated those into spatial and temporal filters so that we can figure out where the active neurons are. And the results are shown here. Here's the raw movie. What our algorithm did was put it into a new representation called SNR representation, where it can de-emphasize bright neurons that are not active. So this is, you can see it, but it's not actually doing anything. So we de-emphasize it in our representation. And it can emphasize dim neurons that are very active, but washed out by the background. So there's actually a neuron here, and we can emphasize that in our new SNR representation. This very much lessened the burden in our deep learning algorithm. And it turns out we can use a very shallow UNet network structure where there's only 3 depths. You can see this is much more simplified compared to our previous network, but it's good enough to do everything, specifically finding neurons in our algorithm. So this was great. Here's some qualitative comparisons. Ours is called SUNS, shallow unit neuron segmentation. It's going to be blue for the remainder of the couple of slides. And the idea is that in a couple of examples, the pale yellow is ground truth, and we can actually find these neurons, whereas other algorithms are not. And in a second example, we can fill out the entire full body of neurons, whereas other algorithms are doing very poorly in terms of filling out the actually human marked ground truth neuron. Quantitatively, we can also assess its accuracy. So basically, recall and precision are marks of reducing false negatives and false positives, and F1 is kind of a combination metric, so it's a catch-all metric. Our algorithm's superior to previous algorithms. And not only that, it was also about an order of magnitude faster than our previous deep learning approach. So now we can process 300 frames a second on a single computer. And that is actually a really big help for neuroscientists as they want to look at their data as soon as it's taken. Lastly, we also developed a algorithm that can process this frame by frame. So it doesn't have omniscience over the entire set of frames for the entire movie, and instead looks only at the recent history. And it performed nearly as well as the kind of bash version, which can look at the entire movie. And it outperformed existing algorithms in this online category, again, by a factor of 3 in speed, which is also faster than the video recording rate, but was far more accurate. So now if we were to process these imaging movies frame by frame, we can actually essentially process every frame on average within about 10 milliseconds. So that leaves about 20 to 40 milliseconds for neuroscientists to actually figure out what the brain is doing and potentially inject some feedback into the brain, which would be very interesting closed loop experiments like the one that Professor Rich Mooney described probably yesterday. Okay, so to wrap up, I've kind of described Lightfield microscopy that can increase contrast and resolution, and I've described these deep learning algorithms that can improve accuracy and speed. And the images actually from my movie, my Ramon and Cajal drawings came from this group outing when we actually went to see the drawings of Ramon and Cajal live. So this picture is actually blown up to 6 feet in our neurobiology building, but it's like the Mona Lisa. It's actually like this big in real life. And the person took it using this microscope. So that the little thing here is the actual microscope that was used to image this thing. And I hope with the additional microscopes that we build, we can provide more detailed and better understanding of the brain. All the folks that did the work were highlighted in bold here. And again, I thank the funding agencies. And with that, thank you. And I'm happy to take your questions.
Thanks. That was a great talk. And we have time for questions. All right, now I see your hand is up already, so go for it.
I was actually going to applaud, I click on the wrong button, but I do have a question. So Yiyang, this is super cool. The SUNS approach. So you mentioned that it's based on a network. So if we try to use that method, do we need to train your network with our data set?
Yeah,
it's already trained.
So it won't be trained. So you'll still need to generate a new network with its weights. But one of the benefits that we see compared to our previous implementation and probably other implementations is that the number of elements in this network is about 5,000. The number of weights in our previous was 2.5 orders of magnitude higher than that. So it took in our previous iteration about a week to train it. Now it takes about 30 to 60 minutes. So as long as you have a decent number of training elements, you can actually train this very quickly within the same day and get a reasonable output, we believe.
Great, thank you.
Could I also ask about that? So when you're training for activity, you're looking at intensity of some fluorophore, right? And then do you worry that by training to a certain, I guess, like a frequency change of intensity, that you're biasing it towards, like if you had a weird neuron or firing at an unexpected rate, that you miss those?
Yeah, so that's a great question. So yeah, I can fill in a little bit about the details of exactly what the temporal waveforms that we use. So let me just go back to that slide. So it turns out we know what the calcium response of lots of neurons are. So when neurons flash, this is the actual action potential. And this is the calcium response. It's a very kind of stereotypical fast rise and then exponential decay, no matter how many action potentials there were. So the temporal waveforms that we expect are these types of fast rise and exponential decay. What we programmed into the algorithm was a filter that matched exactly this waveform and looked for specific things that looked exactly like this waveform in time within the movies. And because of that, If there were other waveforms, perhaps they wouldn't get picked up as well by the sensor. But for the most part, especially excitatory neurons in the brain will have this temporal pattern, and it's very effective in enhancing the signal to noise of those types of features.